Hello students. Students, today we are starting with the 12th chapter of your syllabus, and the name of this chapter is Photosynthesis. So students, in this chapter we will be discussing about everything that is related to photosynthesis, like the raw materials that are used, the pigments that are essential, pigments like chlorophyll, and then there are other certain necessary pigments. Then we are also going to discuss about the type of light, or you can say the wavelength or the quality of the light that is required for the process of photosynthesis. Then there is something called as light and dark reaction, which are the two main parts of photosynthesis. We will be discussing it in detail. And then there are certain adaptations by the plants. Okay, so let us start this chapter by studying what is photosynthesis. Photosynthesis, it is the only process on planet Earth in which the solar energy it is trapped by the green plants and converted into food. And if we see the exact definition of photosynthesis, it is the synthesis of carbohydrate, or you can say the synthesis of glucose from inorganic materials like carbon dioxide and water with the help of solar energy that is trapped by the chlorophyll pigment. So if you see the word photosynthesis, and if you break the word into two equal halves, photo and synthesis. Photo means light or the solar energy, and synthesis means making something or synthesizing something. Correct? So something that is synthesized in the presence of solar energy or in the presence of light with the help of the raw materials like carbon dioxide and water is called as photosynthesis. And this solar energy, it is trapped by the chlorophyll pigment. Now the question that arises is, where is this chlorophyll pigment present? So students, the chlorophyll pigment is present in the chloroplast. Where is the chloroplast present? So the green part or the green leaf of the plant is responsible for performing photosynthesis. So if you take a TS of the green leaf, the upper and the lower part is called the upper epidermis and the lower epidermis respectively. Correct? And on both the sides, the stomata and the cuticles are present. In between the upper and the lower epidermis, there is a mass of cell which are called as mesophyll cells. These mesophyll cells are again of two types. The upper ones are compactly arranged palisade parenchyma cells, and below palisade parenchyma cells there are loosely arranged spongy parenchyma cells. Okay, and if we consider one of this mesophyll cell, a mesophyll cell, it is a typical plant cell. That means it will have a large vacuole, it will have a nucleus on one side and a cell organelle that is called as the chloroplast. Okay, so chloroplast is the place where chlorophyll pigment is present that is responsible for trapping the light. So as said that these are the chloroplast, they are mainly located in the mesophyll cells of the leaf. Now the two raw materials like the carbon dioxide and the water, it reaches the chlorophyll or it reaches the chloroplast through the stomata and through the vascular bundle respectively. So through the stomata, the carbon dioxide will reach inside, and through the vascular bundle, that is the xylem, water will reach inside. Now in higher plants, the chloroplast are discoid or lens shaped. So if you can see the picture over here on the right side, this is a chloroplast. So a chloroplast, it is a discoid shape or a lens shaped structure. Each chloroplast, it is bounded by a double membrane. So there is an outer membrane that you can see, and then there is the inner membrane. So you can see it in the labeling, the outer and the inner membrane. Now inside the inner membrane, there is a ground substance that is found. This ground substance is called the stroma. And inside the stroma, now before that, what is a stroma? Stroma is the matrix, okay, the ground substance, or you can call it as a matrix, or you can call it as stroma. Now inside the stroma, there are certain systems of chlorophyll bearing double membrane sac or lamella present. These sacs, they are stacked one above the other to form the grana. So if you can see this one stack, this is called one grana. Okay? And if you take them individually, so suppose if you take the first one, it is called the granum. And these granum, they are known as thylakoids. Okay? So where are the chlorophyll pigments present? They are present on the thylakoid membrane. Now the function of photosynthesis, this takes place in two steps. The light reaction and the dark reaction. So as discussed previously in the first slide, I said that we will be discussing light and dark reaction, which are the two main phases of photosynthesis. Now the light reaction, it starts when the pigments will absorb the light. That means when the pigments like chlorophyll, carotenoid, xanthophyll, all these pigments will absorb the light. Where are the pigments present? They are present on the thylakoids. So where will the light reaction take place? Of course, on the thylakoids. Okay? Next, the dark reaction. The dark reaction, it takes place in the stroma or in the matrix. Okay, where the pigment is not present. So for the light reaction to take place, pigment is necessary, and for the dark reaction, there is no requirement of pigment. Okay, you will understand the light and the dark reaction better when we take this chapter forward. Now these pigments, they absorb the light of a specific spectrum in a visible region. The pigments are fat soluble, so they are located in the lipid part of the membrane. Now with the help of certain enzymes, they participate in the conversion of solar energy into ATP and NADPH. So where is this process happening? It is happening in the thylakoid. What process is happening? The synthesis of ATP and NADPH. So the enzymes of the stroma now will utilize this ATP and NADPH, which is formed during the light reaction on the thylakoids, to produce carbohydrate. So students, if you pay attention to this, the light reaction where the absorption of light takes place by the pigment, it happens in the thylakoid membrane. Okay, so this process is utilized to form the ATP and the NADPH, that means the energy and the reducing power. And then the enzymes of the stroma will utilize the ATP and the NADPH to produce carbohydrate. Okay, or you can say to produce glucose. And that is how the synthesis of glucose takes place. So as I said previously as well, as we study the light and the dark reaction and all the cycles of light and the dark reaction, you will understand this process better. Now next, let us study about the two predominant types of chlorophyll. Chlorophyll A and chlorophyll B, they are the two predominant types of chlorophyll. Chlorophyll A, it has a methyl group, whereas chlorophyll B, it has an aldehyde group. Now chemically, chlorophyll molecule, it consists of two parts. The head, which is called the porphyrin ring. So this is the porphyrin ring, and then it will have a long hydrocarbon tail, which is called the phytol, which is attached to this porphyrin group or the porphyrin ring. Okay, now this is one pigment. If we speak about the carotenoid being the next pigment, carotenoids, they are lipid compounds which are present universally in almost all the higher plants, and then they are also present in many microorganisms. Now carotenoids, they appear in red color, orange color, yellow and brown color, and they are associated with chlorophyll. Okay, when we say that they are associated with chlorophyll, that means chlorophyll becomes the main reaction center or the main pigment, whereas carotenoid, it becomes the accessory pigments that will be just associated with chlorophyll. It will help chlorophyll to absorb the energy, but it will not take a direct part. It is not the main reaction center. Now, carotenoid, again there are of two types, carotenes and xanthophylls. So the light that is absorbed, or the light energy that is absorbed by these two carotenoids, they are transferred to chlorophyll A to be utilized for the process of photosynthesis. Again, you will understand this better when we study about the cycles of both. Now next, let us study about the nature of light. Now light, it is a form of electromagnetic radiation, and according to quantum theory, it travels as a stream of tiny particles which is called the photons. Photons, it contains a quantum of light, and according to the wave theory, the light travels in the form of waves of different wavelengths having different colors. Okay, so one can see an electromagnetic radiation with a wavelength ranging from 390 nanometers to 730 nanometers. So you can take this as the range. Okay, now this part of the spectrum, it is called the visible light. So where is the visible light present? The visible light, as you can see, it is present in between the wavelengths of ultraviolet rays and the infrared rays. So in between the two, the light that is present is called the visible light, and this visible light is used for the process of photosynthesis. Okay, now let us see about the absorption and the action spectrum. So now we have to understand which pigment absorbs what wavelength. For this, let us study about the absorption spectrum. Absorption spectrum, it is obtained by using spectrophotometer. So after using the spectrophotometer, what we get is this graph. Okay, so this graph, if you can see, it shows us different pigment absorbing different wavelengths. So it starts from 400 nanometer to 700 nanometer, correct? So if we speak about the quality of light, with this graph we can understand that chlorophyll A, that is dark green in color, it absorbs more light in 400 nanometer and 700 nanometer, correct? Same goes for chlorophyll B. Even chlorophyll B absorbs more light in 400 nanometer and 700 nanometer. Now, what do you mean by action spectrum? Action spectrum means when the absorption is maximum, the photosynthesis is also maximum. So this is very obvious that the maximum the absorption of light, maximum will be the photosynthesis. And this interpretation was represented as the action spectrum. Okay, again, what is absorption spectrum? It is the absorption of light at different wavelengths by different pigments is called the absorption spectrum. What is the action spectrum? It is the rate of photosynthesis at different wavelengths of light. Or you can say the absorption, when the absorption is maximum, photosynthesis is also maximum. So if we speak about light, for photosynthesis, it is done in visible light. So as I have discussed previously also, the visible light that ranges from 390 nanometers to 730 nanometers is the optimum range where photosynthesis can take place. Okay, So when we see this visible light, okay, and when this visible light is broken down into certain wavelengths, it was determined that the light is maximum absorbed at two wavelengths, that is 400 nanometer wavelength and 700 nanometer wavelength. So students, I hope that you have understood that this graph. You can see that chlorophyll A and chlorophyll B, it absorbs maximum light in this blue region, that is 400 nanometer wavelength, and the red region, that is 700 nanometer wavelength. Okay, now after this, let us move to the photosystem 1 and photosystem 2. But before we move to photosystem 1 and 2, let us understand about quantosomes and the quantum energy. What is quantosome? Quantosome is considered to be the collection of pigment molecules which help in the process of photosynthesis. So when we speak about a photosynthetic unit, it has one chlorophyll A that is surrounded by the other accessory pigments. So the one chlorophyll A is called the reaction center. So this yellow color structure that you see is chlorophyll A. And surrounding it are the accessory pigments. Okay. And this whole structure or the whole unit, it is also called as the antenna pigment molecule, or it is called as the antenna complex. Now when we say that the light is absorbed by these pigments, the photons are absorbed. So as discussed previously in the nature of light, the photons are something that are absorbed. These photons, they are passed all the pigments, it is passed through all these pigments. So if you can see, the photons are being passed over here and it is reaching chlorophyll A. Same from here, all the photons are passing from all the pigments and it is reaching chlorophyll A. So chlorophyll A being the main reaction center, this is where the reaction takes place. Now if chlorophyll A is isolated, that means there is no other pigment surrounding it, there is only chlorophyll A. Then also the light energy is allowed to fall on it directly. Now chlorophyll A, it gains all the sufficient photons. Now if you see the other picture on the right side. So suppose the light, it falls on the pigment. Okay. And chlorophyll A gains all the sufficient photons and it becomes saturated by the enough or sufficient photons. After that, what happens? Due to enough energy, a pair of electron, it will be excited, okay, and it will just liberate up or move up and it will be accepted by an electron acceptor over here. Okay, so this is happening because of the extra energy that is received by chlorophyll A. Now, as it moves up and it is accepted by the electron acceptor, after that it will move down. It will move down and it will reach its original ground state. So if you can see, there is an excitation light that is of blue color, and when the electron moves down to its original ground state or a stable condition, it emits a different color light. This is called as fluorescence. Now this fluorescence, it is seen only in experimental condition. Okay, it is not seen in living cell. Now, this is just was for you to understand what is quantosomes and what is quantum energy. After this, let us move to the photosystem 1 and photosystem 2. Now, photosystem 1 and photosystem 2, I just want you all to understand one thing. Now, if you see over here in photosystem 1, there are the accessory pigments and then there will be one yellow color chlorophyll A. When the sunlight it falls on the accessory pigments, the photons are absorbed by the accessory pigments and it is passed or transferred to the chlorophyll A, that is the main reaction center, where the main reaction will take place. Correct? Now, due to sufficient photons, or you can say that extra energy which is received by chlorophyll a pair of electron will be liberated up again. That is accepted by the primary acceptor. Correct? Now, the same thing happens in photosystem 2 as well. If you see over here, this is the antenna complex where you will see the accessory pigments. You will see the main reaction center as chlorophyll A, correct? When the sunlight falls on the accessory pigments, the photons are accepted and then they are transferred to the main reaction center. Again, due to extra energy that is received, a pair of electrons will liberate up and it will be accepted by the primary acceptor. Okay? Now you will be wondering that both photosystem 1 and photosystem 2 is similar. What is the difference? The only difference is that in photosystem 1, the chlorophyll A, it will absorb the sunlight or it will absorb the light energy at 700 nanometers. Whereas in photosystem 2, the absorption of light is at 680 nanometers. Okay, so this becomes P700 and this becomes P680. Okay, so this is the main difference between photosystem 1 and photosystem 2. What is the difference? That in photosystem 1, chlorophyll A, that is the main reaction center, will absorb the light at 700 nanometers, and in photosystem 2 it is absorbed at 680 nanometers. So students, after this, let us start with the actual process of photosynthesis. Now this process, it is divided into two parts: the light dependent and the light independent reaction. Light dependent reaction means the light reaction, or you can say the photophosphorylation. Okay, the reason why it is called as photophosphorylation is photo, if you break the word again, photo means light and phosphorylation is synthesis of ATP from ADP. Okay, so that is how photophosphorylation. Light independent reaction is dark reaction. Now when we say light independent reaction or dark reaction, that doesn't it doesn't mean that it takes place only in the dark condition or when there is darkness. It means it does not require light. If light is present, it is fine, and if it is absent, it is again fine. So it is light independent reaction. This reaction will take place in presence or in absence of light. But when we say about the light dependent reaction, or when we speak about the photophosphorylation process, it requires light. Okay, so first we'll be studying about the light dependent reaction, that is photophosphorylation, and then we'll be moving to the light independent reaction. Okay, now photophosphorylation or the light reaction is again divided into two parts: non-cyclic phosphorylation, that is the main process where we get the ATP, and then there is cyclic phosphorylation. So the non-cyclic phosphorylation, it starts from PS2. So as you can see, it starts from photosystem 1. Again, the same thing happens, that the sunlight or the light energy is passed to the accessory pigments and then they are transferred to the main reaction center. Now if you remember in the previous slide, I already told you that the reaction center of photosystem 2, it will absorb at 680 wavelength or 680 nanometer of wavelength. Okay, same thing happens as the main as the main reaction center or chlorophyll A is saturated by the number of photons. A pair of electrons will again go to its excited state, and it will be accepted by the first electron acceptor, that is the primary electron acceptor. Okay, now it will be sent down and it will be accepted by certain other electron acceptor. The first one is the plastoquinone. So the first electron acceptor is plastoquinone. Then it will be passed to the cytochrome complex, and then it will be passed to plastocyanin. Now as it comes down, it will release energy, and the energy that it is releasing, it is used to combine ADP to a inorganic phosphate that forms ATP. Okay, once again, what is happening over here in photosystem 2? As the light energy is passing or it is falling onto the accessory pigments, it is passed to the main reaction center. When the main reaction center gets saturated by sufficient photons with the extra energy, due to this extra energy, a pair of electrons is pushed up to the excited state. At its excited state, it is accepted by the first primary electron acceptor, and then it will be passed down. When it passes down, it is accepted by certain other electron acceptors. The first one is plastoquinone, then is cytochrome complex, and then is plastocyanin. So as it comes down, when it reaches the cytochrome complex, it releases certain energy. Now this energy is used to combine ADP to an inorganic phosphate to produce ATP. So here 1 ATP is produced. Now as it comes down, it is accepted by photosystem 1. So as you can see, from plastocyanin it comes down to photosystem 1. Now as you remember, in photosystem 1, the wavelength is 700 nanometers. Same thing will happen. The energy, the light energy will fall onto the accessory pigments. It will be passed to the main reaction center. Due to maximum energy absorption, a pair of electron will be liberated up. It will be accepted by the primary electron acceptor, and then it will be again sent down. Now as it is sent down, the electron acceptor that accepts it is called as ferredoxin. So as if you can see over here, ferredoxin is the one that will receive the electron, and then this electron is passed to NADP. Okay, so this is passed to NADP. Now this is where it gets reduced. At NADP it gets reduced. Now over here we have to understand one thing, that if the light continuously falls on photosystem 2, that is over here, okay, if the light continuously falls on photosystem 2, how many electrons will be reduced? Because then after it falls on photosystem 2, it will go to the first primary acceptor, then it will come down and it will reach photosystem 1, where it will again be liberated, and then it will be reduced to NADP. Correct? So the question arises is that how many electrons will be reduced? So there has to be a mechanism to replace this lost electron. And this is done due to the photolysis of water. Okay, that is splitting of water. Now, due to the light energy, or in the presence of light, H2O it undergoes photolysis. Now when it undergoes photolysis, it will release a pair of electrons. As you can see over here, when H2O is getting splitted, it will release a pair of electron. It will release a proton and it will release oxygen. So oxygen is released as a byproduct. Okay, so in photosynthesis, if we know, you have studied in the lower classes that oxygen that is liberated is a byproduct. Now, the electron that is due to photolysis, the electron that is produced or splitted, this electron is used as a replacement of the lost electron. Okay, so here the replacement is done, correct? The covering up of the lost electron is done. Then the proton that is liberated over here due to water splitting, it is sent to NADP. So NADP, when NADP, it will accept the proton, it will form NADPH, or you can say NADPH2. Okay, so what is happening over here? We are getting two things in this process. We get the ATP and we get the reducing power, which is used to reduce carbon dioxide in the dark reaction. So students, if you remember, when we studied about chloroplast, I told you that in the light reaction, two things are produced. ATP is produced and the reducing power is produced, okay, which is in the thylakoid region. So where is this light reaction taking place? It is taking place in the thylakoid region, where the pigments are present to accept the light energy, correct? And then the ATP and NADPH2 which is produced, it is sent to the stroma, or it is sent to the dark reaction, where it is used to reduce carbon dioxide or to produce carbohydrate. Okay, now after the non-cyclic phosphorylation, let us study about the cyclic phosphorylation. Cyclic phosphorylation, here what is happening? The same thing. Here only PS1 is involved. Okay, now PS1 will accept the photons. Now again, PS1 will, the chlorophyll A in PS1 will accept the photon, or it will take the light energy at 700 nanometers. The reaction center, when it is sufficient with the number of photons, it will liberate a pair of electrons which will be accepted by the primary electron acceptor. So as it comes down, it will be first accepted by ferredoxin, then it will be accepted by the cytochrome complex, and then it will be accepted by the plastocyanin. Okay, now again over here, when it comes down, when the electron moves down and it is accepted by these electron acceptor, there are two places where the ATP is produced. The first is at the point of ferredoxin to cytochrome B6. Okay, so as you can see, ADP, it combines with the inorganic phosphate and ATP is produced. Then when it moves from cytochrome B6 to cytochrome F, again an ADP is combined to inorganic phosphate and the ATP is produced. Now as it reaches plastocyanin, it again moves to the main reaction center. So over here there is no loss of electron. Okay, there is no replacing the lost electron, there is no loss of electron. In non-cyclic phosphorylation, what we had seen, that there is a loss of electron which is replaced by splitting of water, correct? But in cyclic phosphorylation, this is not the case. There is no loss of electron, because after plastocyanin, the lost electron or the electron again reaches to the reaction center. So in the whole process of light reaction, this completes the light reaction. In the whole process of light reaction, we have studied that there are two things that are produced. ATP is produced and then NADPH2 is produced. NADPH2 is the reducing power, whereas ATP is the energy. Both these molecules, they are helped and they are used in the stroma where the dark reaction takes place, and it is used to reduce carbon dioxide to carbohydrate or for the synthesis of glucose. So students, we are ending this session over here. In the next session, we will be taking this chapter forward where we will be studying about the dark reaction and then certain adaptation by the plants.